Hello, everyone. Let's continue on with our next webinar, presented by Shing-Mou Lee, the experience of development and verification of analytical platforms for cellular and gene therapy, or CGT, products in EMO biomedicine. Click the speaker information tab on the left of your screen for a complete biography on today's speaker. And again, we encourage you to submit as many questions as you'd like during the presentation. We will answer as many as we have time for afterward. Mr. Lee, you may begin your presentation.
Thank you for the organizer invitation. I'm Shing-Mou Lee, the CEO and the lab head of the EMO Biomedicine. Today, I would like to show the experience of development and the verification of analytical platform for CGT product in EMO Biomedicine. Shing-Mou Biomedicine was founded in 2004, located at the New Taipei City, Taiwan. Our main focus are analytical and manufacturing platform for CGT products and cell-based bioassay. Current business activity, including testing and manufacturing service for cell-based products and contract research service for biologics. Regarding the manufacturing service for cell-based product, EMO's cell processing unit, CPU, is in compliance with good tissue practice guidance and certificated by Ministry of Health and Welfare of Taiwan for the processing, culture, and the storage of the therapeutic cell-based products. It means under the special regulation on cell therapy in Taiwan, EMO can provide the certificated product to hospital for treatment. For the testing service for cell-based product and clinical specimen, EMO's testing lab is in compliance with ISO IEC and CNS and accredited both by Taiwan Accreditation Foundation, TAF, and the Taiwan FDA. The testing scope includes whole blood, cell culture medium, suspensions, supernatant, biologics, and the cell-based product. The safety test for cell-based product and biologics, including mycoplasma testing and the verification and the endotoxin and the sterility test. The safety test follow the pharmacopoeia and are accredited by TAF. For the identity for the cell-based product and full blood, including lymphocyte and MSC stem cell. Most of the gene-modified cell-based products are customized. The functionality and the potency development development service, including the T cell and the MSC. We will talk about the idle quantification assay later. EMO is also a qualified CRO by Ministry of Economic Affairs for biopharmaceutical company since 2013. EMO specializes in cell-based bioassay and the clinical specimen preparation. And recently, the development of the gene-modified cell include CAR-T, NK, and the TCR-T increasing very rapidly. So to meet the more and more testing demand, EMO has preparing introduced the digital PCR for RCL, VCN, and other testing. Up to date, EMO has established one-star service including manufacturing and testing for cell-based product and bioassay. We would like to provide this service to local and overseas clients. Today, I would like to show two experience in our lab. The first one is method verification of mycoplasma detection for CGT products. Mycoplasma testing is necessary and unique safety test for lot release and in processing control for all cell-based therapeutics. There are three mycoplasma detection methods listed in Taiwanese pharmacopoeia and European pharmacopoeia, including cell culture method, indicator cell culture method, and nucleic acid amplification method, so-called NAT. According to the guidance for NAT method validation and verification for mycoplasma detection in European pharmacopoeia and Taiwanese pharmacopoeia (TP). If we want to replace the culture method, the NAT system must be shown to detect 10 GFU per mil for 8 species of mycoplasma described in TP or EP. The commercial kits are already validated by the kit manufacturer and replace validation by the user. However, the performance of the kit with respect to its intended use has to be demonstrated by the user. That is method verification. When user perform mycoplasma detection with validated commercial kit. Authority in Taiwan, Center for Drug Evaluation, CDE, recommends to run method verification in the following three scenarios. The first one is completely follow the instruction of the kit. User only has to demonstrate that the detection limit reaches 10 CFU per mil. And the second one, using other brand DNA extraction kit with the same principle or real-time PCR, then user has to test concentration other than 10 CFU per mil for the robustness. The third, when using different principle of DNA extraction kit, and the user has to revalidate the method because it is a big change. We compare the detection limit and the robustness in this table between the method validation in Taiwan Pharmacopoeia and the method verification recommends from the Taiwan authority CDE. For the objective, the validation has to establish the detection limit, and in the verification, only has to confirm the detection limit reaches 10 CFU per mil. And for the number of the mycoplasma validation asks to test 8 species, and the verification only asks the user has to choose at least 2 species of mycoplasma after risk assessment. For the concentration of mycoplasma, TP validation as tenfold dilution series, and the verification only as 10 CFU per mil. Number of replicate is the same, and the result interpretation, the validation as the concentration of mycoplasma that can be detected in 95% of 24 replicates will be the detection limit. In the verification, mycoplasma with 10 CFU per mil must be detected in 95% of 24 replicates. And this is an example of validation result conducted in immerse lab. We follow the recommends of the CDE. You can see we test at three species of the mycoplasma species, and the 3 independent tests, and the positive detection rate over 95%. The following, I would like to show our second experience. The assay development for potency assessment of MSC products. A potency assay must reflect the product's mechanism of action and correlate to the safety and efficacy data generated in preclinical model and clinical study. Establishment of a potency for MSC product has been a challenge due to the issues of variability in studying the raw material and the complex mechanism of the action and lack of reference cells or materials. The following slides based on our two publications in cytotherapy related to the assay, potency assay development and phase 1 clinical trial. And a quantitative flow cytometry-based assay standardized by the fluorescent intensity of beads was developed to measure idle in interferon gamma prime MSC. Idle is one of the most important factors for human MSC-mediated immunosuppression. The immunosuppression was assumed to be the MOA of MSC product, Rexstat, for the OA treatment. The quantitative idle assay for measuring immunomodulation of interferon gamma prime MSC product was applied in the manufacturing process of phase 1 trial for osteoarthritis treatment. A flow cytometry intercellular blending method used to analyze the ideal positive percentage of the cell population in adults. The ideal IPV value represents the average relative quantity of idle expression in a single idle positive cell. Two kinds of the in-house quality control ideal produce reference cell were running together with the MSC product to ensure that the assay performing its aspect. We analyze the idle IPV value of 12 MSC products. We observed the individual variability of ideal IPV of 12 patients' MSC products. It shows the study materials variability. We noticed 3 patient MSC products S04, 09, and 12 with significant low ideal IPV, the 04, 09, and 12. So we analyze the correlation of ideal expression level with clinical effects of OA treatment. We found the expression of idle or interferon gamma prime MSC positively correlated with the pain relief in the clinical trial for OA treatment is stable. And the 3 low ID or IPV patients S04, 09, and 12 in the trial with worse pain relief results compared with the average of 9 subjects in the trial. And we also check the inhibition of macrophage TNF-alpha secreting assay. We found MSC product with low idle expression has less capability to inhibit TNF-alpha secretion of activated macrophage. And we also analyze the ideal versus the other biological assay. The most popular assay is suppression of T cells. And we found the positive correlation between quantitative idle assay and the inhibition of T cells proliferation assay for MSC products. It's the summary of our study. The quantitative idle assay used resting MSC as an internal control and the 2 cell line as reference materials to monitor the whole process of operation. Idle is expressed in interferon gamma prime MSC, but not in resting MSC in vitro. The quantitative ideal assay can meet the general requirement of validation, accuracy, precision, specificity, linearity, and robustness, and the major immunosuppressive capability of MSC. The quantitative idle assay is applicable for comparability, compatibility, and stability study in MSC product manufacturing. The results of the quantitative idle assay has high correlation with T cell and macrophage suppression bioassay, as well as clinical efficacy. Thank you for your attention.
Thank you, Mr. Lee, for your informative presentation. Mr. Lee is unable to join us today for the live Q&A segment. However, all questions asked during the presentation and also during the on-demand period will be responded to via the email address you used at the time of registration. Before we end this webinar, I would also like to encourage you to fill out the session survey that's found on your screen. Our next webinar, titled Safety First, a comprehensive look at cell and gene therapy testing, will begin in approximately 5 minutes. To enter that webinar, all you need to do is close this window and then click on the button for that next webinar found in the auditorium. We'll see you shortly in the next presentation. Thank you.